thank you very much indeed. It's a great pleasure to be here, and what a great act to follow. I hope I can be as invigorating. I'm going to talk about aging resilience. I want to introduce you to that idea. Older people are really very varied from each other. All of us know 80-year-olds, some of whom might be really very fit, or some of whom might be much more frail. And I wonder whether we can understand that difference between people of the same age to really get an understanding of how we might be able to level things up for people who are a little bit more frail in aging. Before we start, I just wanted to talk a little bit at first about the difference between age and aging. So here we've got a graph of grip strength. Grip strength is a really good marker of your psychomotor performance, and it changes with age. You can here see Bridget, one of our nurses, who's doing a test with one of our twins. And this is a plot. Each little dot is an individual measurement, and it's plotted against age. And unlike the straight line in the diagram, you can see that actually there's a bit of a curve. So so men increase a little bit up to their 40s or 50s, and then readings start to decline. In women, it's the same but a little bit lower level. But the issue is that these individuals here that are in pink, they have grown up and been exposed in very different times. These people would have been born just before the First World War. So their entire early childhood, they would have grown up in wartime. So it's a bit difficult to then, do your science where you compare those older people to much younger people, because you might be looking at things that are not just aging. They might be related to things that have happened to them over time. And so what about if we look upwards and downwards in this graph? So we look about the difference between people of the same age, or even if we take one of those dots and we follow it over time, how it wobbles, how it changes, and how it changes over time, perhaps declines as well in later years. The reason why I'd like to do that is because actually I think many of us want to understand intra-individual aging resilience. My kids were pretty horrified at these pictures here, but I found it quite fun to make them. But basically, these are two different versions of future me made by AI. And what happens if, they look quite similar, but what happens if actually we have some event like I fall over? Well, one of the versions that might look quite similar to the other might then have enough time to put out their hand and save themselves from major injury, but just fracture their wrist and get over it. And the other might not have quite so much time and fracture their hip and then go on to get a series of other events and then die. So these are the things where small differences might make a difference. And we need to understand what we can do to change that future for all of us individually. So that's why I went to Tim's department to study twins, because twins share genetic factors and things in their early life, and these are things that we as adults, we just can't change. And so studying them might be able to tell us more about the modifiable factors we can act on later in life. And one of the first questions I wanted to ask with twins is to really understand what's the impact of genetics on aging. And what we found is that actually genetics really is quite strongly responsible for that top peak of that curve that we saw, for example, in grip strength. But actually then the changes over time after that are really not driven by genetics. They're much more explained by environmental factors. And then, so was there one common factor, environmental factor, which was altering all these different organ systems? So we looked at the heart, the lungs, the skeletal muscle system, the bone system, and metabolic health. And what we found was that instead of one common factor, we had an extremely complicated model. So here, A is genetic variant, C is shared environmental factors that's shared between twins, and E is non-shared environment. And we saw that there's a lot of arcs, actually. There's a lot of common shared variants between all of these traits, but no one single factor. These different systems all age differently, and the shared components are really largely explained by environments, not genetics. Now, it took about 8 years, actually. That was the first paper really looking at trajectories of aging in different organ systems. It took about 8 years before anybody else replicated it, but I was very pleased they did. So this is the first paper was from the Framingham studies, showing that trajectories in different organ systems measured through physiological measures or clinical measures are showing different aging trajectories. And in this paper, which was from UK Biobank, they showed as well that the environmental factors are really key, and they tried to identify some of them. And in this one, we saw that biological markers were changing differently in different body tissues, but also different cells. There are separate organ system aging, but these tap also different aging associated processes. And so here we've got the 12 hallmarks of aging that work upon cells, and indeed all of these factors might be happening differently in different cells of your body. So ultimately, our body systems age at different rates, and so this might mean that we need to approach things differently for different systems or for different individuals. And there could be that there's trade-offs. So in other words, if we help one system in the body, we might have trade-offs. It might not help another. But these are questions I'll leave for a moment, because what I set out to try and find out after finding out this was really what are these shared environmental factors? Because if we can target those, maybe we can target something core, something that will then be translatable and make a difference for lots of older adults. So you can see intra-individual aging. It's quite complicated. It's non-linear, it's multimodal, and it's multidimensional. And actually, that's very much the same for my research course, life course as well. And obviously, there's lots of tributaries, lots of people that have contributed over the time. There's a lot of wobbling and meandering about from one subject to another, and there are multiple possible futures in the end. And so in this lecture, I'm going to talk about 3 critical influences in my research course, life course. But I'm also going to talk about 3 contributions, hopefully, that I've made so far to the understanding these life course influences on aging trajectories. And also talk about how we might think about creating that future landscape for other rivers to flow. And I think that's a very important part of what we've got to do as professors at King's. So here we go. Now, I was born CJD, which was a difficult, so I was Claire Joanne Davis, so it's a bit of a difficult set of, what's the word, abbreviations to grow up with in the 90s. And I grew up on the River Sow, which is up in Staffordshire. It's a tributary of the Trent. And I like the river analogy because actually we're, as I've said, shaped by people and by the landscape in which we grow and live. And that's the same for our research courses. And 2 really important influences in my early life, these 2 tributaries, I guess, that formed me. My mum and dad, they're here today. Really amazing people, doctors, really brought me up with the idea that we were here to make a difference to the world. They also gave me so many resources and opportunities, opened every door for me, and really gave me so many advantages, which have allowed me to take this stage today. So I'm super grateful to them. They've always influenced me and still do today in every decision I make. So thank you, Mum and Dad. And here's Franklin. This is my husband. And he comes in really very soon. You can see this little village of Chebsey. That's where I was grew up. And we met, actually, 32 years ago. We've been together for 31 years, and he is my soulmate. I'm very lucky that I met him so early on, because having that bedrock of a person alongside you in life is so important. And he drives me to fulfill myself in my work, and I hope I do in him as well. And he also played a huge role for me in COVID pandemic, where he looked after our 3 children, whilst I was working on the COVID symptom study app. And then also, alongside that, he's really doing very impactful work himself, which I'm sure he'll be happy to tell you about. So thank you. Thank you so much. And actually, we met before I even went to university in Cambridge in 1994, and went to study medicine. And after I did a bit of medicine, I actually then did a degree, or a sort of integrated degree in history and philosophy of science. And here's a nice mugshot. I was trying to look very serious here because I didn't want any Russians not to let me into their science libraries, to do a load of archival research around this question of abortion in revolutionary Russia, 1905 to 1920. So I wasn't afraid of a serious subject. But actually, it was the first time that any country in the world had legalized abortion was in 1920. And there were lots of debates and fierce sort of like discussions going on in the build-up to this. It was apparently a pressing social and health problem. But in fact, when I looked into it, there was absolutely no data that this was an issue in Russia at the time. All the data was imported from the UK and the USA. So in fact, policy was being formulated in a data vacuum. And so two things that I really learned from this was, first of all, that understanding really comes when you investigate why things are not what you thought they were. So the things we get wrong are much more interesting than the things that we get right, that we can predict. And really, data is important and helpful in making good, lasting decisions in policy making. So then I went through, obviously, the medical course, but I heard about the Jarman Index, which is the precursor of the Index of Multiple Deprivation. And I realized that if you want to study medicine, it's probably a good place, good thing to do, to go to places where there is a high level of deprivation. So I moved to London to go to the Royal London Hospital, which at the time had the highest Jarman Index. And actually, I wrote to Irene Higginson and said, can I come and work with you? And she gave me a small project over the summer, which was my first piece of research, doing research in older people. I'm very grateful to her for having done that, because it really put me on a different path. And I think all of us, when we get emails from students, it's really good to respond to them and to really engage them, because it could make a really big difference. The other person who made a big difference to me in those medical school time was John Moore-Gillen, who we did a project, again in East London, looking at tuberculosis in a population study. And then I went and became a junior doctor. And much like other junior doctors now, it becomes a bit difficult, and it's quite difficult to find your way sometimes. You try out a lot of things, and you don't really know where you're going. And in fact, we then went off on nearly a year going sailing to find our own course. And here's our little boat that we still have. It's a fantastic thing to get away to. And it was actually on that boat that then I realized that actually I really enjoyed looking after older people, and I found older people most exciting. So I decided to come to geriatric medicine. And here are the inspirational, some of the inspirational, actually there are many more as well, but here I can only fit 4 on the slide. Inspirational course setters in geriatric medicine here in GSTT. And these are people who are really proactive in designing real solutions here and now. And you can't see it very easily, but this is a peace document. So this is the Proactive Elderly Advanced Care Plan, which I developed as a registrar, together with Finbar Martin, who's here, back when I was a registrar. And I looked online, and actually it's everywhere across the south of the country. Here it is from Dorset, and I think it's in South Essex. People are using this to this day to make an advisory advanced care plan for people as they leave hospital. But then I went to do a PhD, which I did with Tim Spector, who's here, this mugshot here. You can see him in orange standing next to me with the twins at this time. But you can also see Debbie Hart, who's there, and various other people that are still in our group. And what I did was I used some work from Raj Gill. I'm just see if I can get the pointer to work. Raj Gill here had done some cognitive measures 10 years before, and I was looking at the changes in those cognitive measures, what predicted them. And I'm also extremely grateful to my main supervisor, who's Steve Jackson here, and he's here depicted with Julie Whitney, who was a previous PhD student of his, who was really instrumental actually in helping me, and also being a role model as a mother as well of 3 children. But here what we showed actually was that physical fitness was the really key driver of longitudinal aging in your cognitive system. And in fact, you don't need to be a neuroradiologist to see that these are 2 twins. The brains are extremely similar, but this one, there's less black space, there's much more brain volume, and there are differences in both brain structure and function in twins that have different exercise exposures. So I think this is really important, and it's very much sustained by other work, that physical fitness, of course, is one of those things which might affect multiple systems at the same time. But there's another thing as well. So Tim was starting to work on microbiome. Together with Jordana Bell, they set up a project with Cornell University where we looked at microbiome in relation to lots of different diseases. And so I came on the shop floor and wanted to understand whether this was also related to frailty. And we showed really convincingly that actually robust twins had much more of these helpful bacteria that seem to be causally associated with reducing inflammation in mice. And they're really highly networked species which produce molecules which feed the community of microbes but also help the gut be healthy. And these are associated with more robust twins. And then my PhD student, Matt Jackson, this is two Nature Communications papers that I want to show you sort of a story arc. They're actually quite 6 years apart in publication. They're published in the same journal. This one's done by, the second one's by Mary Nee Loughlin, published a couple of weeks ago. But what Matt found was there was these health-associated microbiome across loads of different diseases, which correlated with plant fiber intake. And in fact, one of the key bacteria that seemed to go up was Bifidobacterium species. So we tried to look to see whether or not we could do something to alter that and see whether that would actually change both your strength when you stand up and also your cognitive function. And this is Mary, who is she was my PhD student. She's now Dr. Mary Nee Loughlin. And she showed that if you give the twins that you give this prebiotic food supplement to had better cognitive function after just 12 weeks than if you gave a placebo. It was a really nice, randomized, neat design within twin pairs. I'm so really pleased that we can sort of see that story arc of something being discovered and then something being actually put into practice so people can change their outcomes. But also there's other things that we can change outcomes in older people in relation to microbiome. This is C. difficile infection, which is a pretty nasty infection that older people get. And we know that fecal microbial transplant is a kind of rescue procedure and is mostly used in young people, actually. But this amazing young geriatrician in Denmark, who I'm a third supervisor for her PhD, she's done a project where she's given fecal microbial transplants to really frail housebound people and found actually that they really improve in function alongside a comprehensive geriatric assessment. And that paper's actually not yet been published, but it's been submitted for review. So now I'm going to come to the next bit, because around this time, COVID hit. And it may not have been a revolution, but obviously we realized that we really needed data. And this was an amazing sort of collaboration between Tim, who has such amazing good media relations and could get out to 4.6 million people in the UK in such a short time. Me, I was working on in care home, trying to find actually a point of care test, early point of care test for COVID, but identifying people with symptoms. And Seb Orsalan, who had this amazing data science structure, together with Zoe, we put together in 4 days this app, which I'm sure you mostly know about, but we managed to do lots of things that then impacted on progress. So first of all, using data, obviously people knew that sense of smell, quite early there was reports that sense of smell was an issue, but we were the first people that really showed the data that actually changed not just UK policy, but WHO policy. Secondly, we were the first people that showed the data that showed that delirium, acute confusional state, affects it could be one of the only symptoms presenting in frail older adults, and we changed PHE guidance immediately after this paper with a lot of help from Age and Aging and the British Geriatric Society. And we were one of the first people to really describe long COVID with one of the, well, certainly the first controlled study showing what the attributes and predictors were of long COVID incorporated into the NICE guidance. Later on we looked at the development of illness after vaccination, and we were able to contribute to booster vaccination campaigns. So there was lots of papers, the time series data was super useful, and we got through this a direct line to the Chief Medical Officer, UKHSA Vaccine Task Force, and Twins UK actually became a Vanguard national core study. So at the same time as doing the COVID app, these remarkable individuals, Ellen Thompson, Ruth Bowyer, and Nathan Cheetham, were working with me together with 12 other universities in the UK on the national core studies, bringing together all these national longitudinal population studies to understand COVID. And we did lots of really fantastic stuff. And I want to bring us to this sort of like third element that I was talking about things that influence multiple system aging. And really the important thing is inequalities in, inequities in social factors. And so here, in this paper, which actually is our most cited paper so far, it showed that healthcare workers had a 12 times increased risk of getting COVID in the UK, but only a 3 times risk of getting COVID in the United States. And that was driven by practices on using PPE, because in hospitals at that early stage in the pandemic, we only used PPE with people we thought were infected. We didn't use PPE in a situation like this or in the ward round sort of huddles or whatever. And actually, I think this paper led to a change in PPE policy directly after its publication in UK hospital settings. But the really sad thing is that people of racialized minority racial groups had reduced access to PPE, but also people who were working in care homes had reduced access to PPE. These are the sort of yellow spots here compared to in hospital settings. So there was racial and spatial inequalities in PPE provision. Then is another inequality in prevention, where and how you live. So Ruth Bowyer did this paper together with Christina Meni, which showed that there are really strong geosocial gradients in who gets COVID. And again, we were the first paper to publish that before even the ONS came out with it. Michaela Antonelli showed that you're more likely to have a post-vaccination infection if you were very frail, but also if you lived in high deprivation areas. And then Nathan Cheetham showed that antibody levels, actually in high deprivation areas, there were different responses to the vaccine, and that might be partly what explained that post-vaccination response to infection. And that was explained to the vaccine task force, and that led to lots of pop-up vaccination centres. I mean, obviously they were doing them anyway, but we were strongly advocating that they would pop those centres up in high deprivation areas and have visits for housebound, frail, older people. But despite this, unfortunately, we've seen in a recent paper in The Lancet that we did a commentary on together with Martin Gulliford that 75% of the avoidable severe COVID cases because of failure to fully vaccinate are in the over 75. So that's where we need to be going to increase our output rather than chasing younger people who are vaccine avoidant. And finally, sort of about inequalities of mental health. So this is work really mainly by Ellen Thompson, who, together with 12 other longitudinal population studies, really showed that actually the disruption to other healthcare was much more likely in people with mental health problems, together with women and ethnic minorities. So disruptions to healthcare during the pandemic have widened social inequalities in health. And also, Nathan showed that recovery from long COVID was associated with strong social advantages. So now what we're doing together with these fantastic people over there on the right, we're doing a study to really understand. So we know that social factors are really strongly indicative of what might happen in response to a stressor. But how does the biology of that work? That's what we're trying to understand within this Wellcome Leap funded project, where we want to identify the biology behind the reaction to this stressor using the twin cohort, which we've assayed all through the pandemic. So I'm just coming back to the sort of rivers idea, and I wanted to just reflect on how all of this is only possible in the context of a research environment which is really fostering. And I'm super grateful for everybody in the School of Life course and Population Sciences within the whole faculty, but in particular people in research management and innovation, so the contracts team and so on, who really make the research possible. And indeed, in all the grant applications and so on, the research strategy and development team here at King's. And I really want to think about how I can help to build our research environment going forward. And in my view, the pandemic may be over, but the threats are not. And we have two major threats, environmental change and frail aging, coming at us right now, population aging. And how can we use science to really understand how older people can bounce back from these stresses? And we've got, thanks to Folsom funding, we've got a new Centre for Aging Resilience in a Changing Environment, where we want to be able to bring all the attributes together in King's to understand this. And here are some of the things that we've, some of them have already got funded, which are sort of like thanks to this collaboration. So there's the thing that I've just told you about, the LEAP thing, but also this is Carly Welsh, who's leading this incredible project where we're looking at older people undergoing surgery. We've got Wendy Hall, who's leading a project around sustainable health and aging nutrition. We've got Daniel Schillerif and others in geography who are working projects up to look at indoor environment and health quality, together with others that are here, like Josh Foster, who's looking at heat stress. We've got a new project that we're doing together with Richard Dobson to looking at health research from home, and I see Will Dixon is here as well, and this is a project that we're doing with lots of collaborators all the way around the UK. And any of you who were at Jagdeep's lecture a few weeks ago will know about her amazing project with Jude Partridge, which is going to be looking at how they implement POPs up across 18 centres around the UK. But there are 17 seed funding projects we're meeting tomorrow to discuss, where we're trying to come up with solutions for both net zero and ageing resilience. I'm really excited about that. But I did also say that there was a third, critical influencer for our research. And I really think that these guys are the third influencers. They are rocks of our research, and they're our team at Twins, the professional services team led by Debbie Hart here. And I want to really say thank you to them, but also thank you to these incredibly supportive peers and colleagues, to Tim Spector, who's really been my mentor through all of this, but all of us working really closely together in a really tight-knit team. Really thank you to everybody, and thank you to the Twins.
Fantastic. Thank you very much indeed, Claire. Professor Tim Spector, vote of thanks.
Thanks very much. Two fantastic talks. Just wanted to say that Claire came to my attention really because every time I passed one of the geriatricians in the corridor at St Thomas's, they say, you really ought to have a look at this registrar of ours, Claire. I think she's great. Sure, she's going to be a future star. And I kept ignoring it, but each time one of them passed me, they'd always mention her. So it wasn't a surprise then. We did have a meeting, and Claire decided, yes, she would do a PhD, but she never had more than about 2 minutes to discuss it with me because her bleep was going off. Or there was some problem with one of her children from the school, so there'd be always some phone call. And it was actually amazing that she did come and do that PhD, which was incredibly ambitious, like most of the things that Claire took on. Learning how to do MRI scans and going around and dealing with twins. And it was about 5 projects in one, really. And that really typified her enthusiasm and energy. And I really didn't see much of her. I think it was a sort of DIY PhD, really. We just used to really sort out the problems, and I came in when it was, how do you make some papers out of this stuff? But it was all incredible, and she was just so hardworking, so full of great ideas, and just kept having children, which was not. Totally amazed me how she managed to do all that at the same time. But I didn't really, I don't think, get to know Claire until COVID hit. And I think this was an amazing time that when we got together, had this idea and created this app together with Zoe in 5 days that I think will be the lasting legacy of our work. And if we look back, in 20 or 30 years' time, say, well, inventing that app that helps so many people, so many millions of people, I think is an amazing legacy that we'll all be proud of. And it came about because we were able to work as a team, and because Claire is such an amazing person, not only with her enthusiasm, her skill, her brightness, her intelligence, but she's got this empathy and this way of getting people to work with her that is incredible. And I think that's something that I still marvel at. And she got all these disparate people, and she found Seb Orsolin and I would never met, and put us together and said we ought to get this together. And I think it is incredible. And we look back, these 4.5 million people that were using this app, and people still coming up to me and said, thank you so much. You really helped me through this. And I think it was largely due to Claire's drive, enthusiasm, and the fact she was working on the front line when we did this, still taking incredible risks, getting samples in those early days about how to get people's COVID tests sorted out. And I think really together, we did change history, and I think we should be very proud of that. But it was, and that's really how I think we got to work so closely, and it was an amazing period of my life, and I think it was due to very much to Claire and her force of personality that made it all possible. And so I was a bit nervous about scaling down a bit at King's and handing over head of the department of twins, which I'd set up 30 years before. It's giving up your baby, really, for adoption. I said, well, Claire, how many, 6 months, 12 months, 18 months, when do you want to start? So I'll start next week. I was, all right then. And I was a bit nervous. But she's taken over, and you can see she's suddenly got 3 massive grants and everybody working on her side, huge plans. And really, I don't know why I didn't do that before. So I'm very happy that she's going to do a much better job. And as you know, she's far better at many things than me, and particularly with diplomacy and getting on with everyone else. So, Claire, let's raise a glass later on to Claire and Elizabeth, 2 fantastic new professors. Thank you.